So the elementary canal. So we are beginning from here. Elementary canal in the members of platyhelminthes has only single opening to the exterior, and it serves as both mouth and anus. Let's take a representative from there that to understand. Fasciola hepatica. In case of fasciola hepatica, if you see that fasciola hepatica, which is called liver fluke, a representative of phylum Platyhelminthes, you see here, this opening acts as both mouth and anus. Here it acts as both mouth and anus. Okay, so let me show you once that phylum Platyhelminthes is example for sac-like gut. That it is said to be example for sac-like gut. Okay, next from phylum Nematoda to Chordata. From phylum Nematodes to Chordates, if you take a roundworm as an example, Ascaris, a representative from phylum Nematoda or Aschelminthes, that you see the elementary canal begins with mouth and ends with this is mouth and this is anus. Elementary canal begins with mouth and ends with anus. Okay, so based on the gut, we said there are two types. One is sac-like, another is tube-like. Sac-like gut, which contain only one opening, that only acts as a mouth and anus, is there in phylum Platyhelminthes. And the tube-like, which begins with mouth and ends with anus, is seen in the case of phylum Nematoda to Chordata. Okay, so that's it about sac-like and tube-like gut. Next, the blood vascularization, open type and closed type. Open type and closed type. So let me show you that open type and closed type of blood vascularization. If we come to the human beings, that if I take the heart like this, from the heart, the blood vessels will come and they get capillarized into that small capillaries, and all the capillaries again becomes the blood vessel, and that they open into the heart. There is no opening at all. There is no opening at all. We say such kind as closed type of circulation. Such kind of circulation is called as closed type of circulation. In this type of that blood is circulated through a series of vessels varying diameter. That means that in the diameter point of view, this is called artery. Okay, so diameter artery, and these are veins, and in between we'll find capillaries. So arteries, veins, and capillaries. This is artery, and this is vein, and box I am showing that is capillary. So we call such kind as closed vascular system. In case of closed blood vascular system, you'll find annelida and cephalopoda, cephalochordates, and vertebrates are exhibiting this kind of closed type of circulatory system. So that let me show you once again. Let me read. In this type, the blood is circulated through a series of vessels varying diameter, that arteries, veins, and capillaries. So where do you see this? In case of annelids, cephalopods, and cephalochordates and vertebrates are having these kind of closed type of circulation. This kind of closed type of circulation is seen in them. Next, what is open type? We'll take cockroach from phylum Arthropoda as an example. In case of cockroach, that we'll find that the 13-chambered heart is present here. 13-chambered heart is there. So I'm not showing the clear view of that, just showing that how does the open vascular system is seen. So, in that anterior, the first chamber is having a blood vessel. We call it as aorta. Okay, so from the heart, that blood is pumped into the body cavity. There is no capillaries here. There is no blood capillaries. The blood is going to be pumped into the capillaries, vessels, the sinuses, body cavities, we say the sinuses. So no capillarization. Okay, so let me read that in this type, the blood pumped out of the heart flows through open spaces. We are talking about the sinuses, bathing the cells and tissues directly, we say open type of circulation. Blood vessel is present. This is the blood vessel. Blood vessel is present, but capillaries are absent. So that is open type of circulatory system. So from this particular area, what we learned, the sac-like gut of phylum Platyhelminthes with only one opening which acts as both mouth and anus, and tube-like gut, alimentary canal begins with mouth and ends with anus. Sac-like gut in phylum Platyhelminthes and tube-like gut is seen in phylum Nematoda to Chordata. Then in the same way, that open type and closed type of vascular system also we are seeing here, that in the open type, blood pumped out, the heart flows through open spaces, bathing the cells and tissues directly. Example, the members of Arthropoda, Mollusca, Echinodermata, and Urochordata. In case of closed type, the blood is circulated through a series of vessels varying diameter, artery, vein, and capillaries. Annelids, cephalopods, cephalochordates, and vertebrates are having these kind of vascularized system. That this kind of vascularization is present there. Okay. During the evolutionary history of animals, there has been a tendency towards increase in the body size. During, we have a question here, during the evolutionary history of animals, there has been a tendency towards the increase in the body size. Explain why. Here the question is asked over. With the basic information, there are so many reasons are present. With the basic information, we can say, whatever we are, according to the chapter, we can say, earlier, unicellular organisms are present, single cell. Then after what happened? Multicellular. Then after a certain group of tissues are getting formed here. And tissues are organized into organs. Organs are organized into organ systems. That means when we are going from a single cell level to the organ system level, definitely in the due course of time, the size of organism will increase or not. So in that angle, the question is asked over, our answer is also given in that angle. That means protoplasmic grade of organization, cellular grade of organization, tissue grade, organ grade of organization, and finally we reach to the organ system grade of organization. In the due course of this kind of evolution, definitely there is an increase in the size, isn't it? Then next, we are moving towards diploblastic organization. Diploblastic organization. Okay, so from here onwards, that we need to have that idea on our kingdom animalia chart. The kingdom animalia chart is going to be must for us. So for that, I'll make you open that kingdom animalia chart also. See, once I'll say once again, kingdom animalia classified into the two subkingdoms, subkingdom Parazoa and subkingdom Metazoa. That we have completed this now, cellular grade in subkingdom Parazoa with phylum Porifera, and tissue grade in case of grade radiata Diploblastica, and organ grade and organ system grade, both we will find in case of Bilateria or Triploblastica. Now we are here, Diploblastica. So what is Diploblastica? Diploblastica means the presence of two germ layers here, that we learn basic line as two germ layer, ectoderm and endoderm. Then the thing to remember over that animals showing the diploblastic condition, they're also called radiata. That means they also exhibit radial symmetry and they also have tissue grade of organization. These kind of interlinking is required for us. That whole and soul example for all these two germ layers, radial symmetry, tissue grade of organization are phylum Nidaria and Ctenophora. Phylum Nidaria and Ctenophora. Okay, so that's why I'm saying that kingdom Animalia, knowledge of kingdom Animalia is must for us, that make us to get perfection in this area. Okay, so let me go for the chapter back again. Let me go for the chapter back again. Let's start the diploblastic organization. In case of diploblastic organization, we said already there are two germ layers are present. Let me read here. Animals in which the cells are arranged with two embryonic layers. One is external ectoderm and internal endoderm, which we call them as diploblastic animals. Example, phylum Nidaria and phylum Ctenophora. Nidarians and ctenophores are exhibiting this particular phenomenon. The ectoderm gives rise to the epidermis. Remember that ectoderm, that means that is embryonic germ layer. Ectoderm. During embryonic development, we'll find that layer. That means that ectoderm, which is there in embryo, what he is saying that will germ that develops into epidermis. In case of adult, the ectoderm develops into epidermis. Endoderm gives rise to gastrodermis. This endoderm, whatever there inside, that gives rise into gastrodermis. So in adult, in embryo, we say it has ectoderm and this is endoderm. Okay, so in adult, when it move, we need to use, let's take an example that hydra, that in hydra, outer layer is epidermis. What is the inner layer? Inner layer is gastrodermis. Inner layer is gastrodermis, the tissue that lines the gut cavity. This is the gut cavity. The cells in each tissue layer are functionally independent. In each tissue layer are functionally interdependent. What do you say? The cells in each tissue layer are functionally interdependent. That was the change actually. In case of phylum Porifera, we said there is no coordination. That's why they are independent. Here they are interdependent. Tissue grade of organization, we can say. An undifferentiated layer present between the ectoderm and endoderm. That undifferentiated layer present between the ectoderm and endoderm is called mesoglia. I am showing here that mesoglia between the blue and red. I am drawing yellow. So what do you say that as mesoglia between the ectoderm and endoderm? Mesoglia is present. In some diploblastic animals, cells occur in the mesoglia. That means, in case of higher cnidarians, we'll find the cells are present there. Cells occur in the mesoglia, but they're always derived either from ectoderm or endoderm. So we are saying that some cells may be present. In case of class Anthozoa, we are going to learn in next chapter. We'll find some cells. In case of Scyphozoa, some cells are there. In case of Anthozoa, some tissue is there. But what they're saying, but they're always derived from either from the ectoderm or from the endoderm. Okay, so this is the story of diploblastic organization, as in which the cells are arranged in two embryonic layers, external ectoderm, internal endoderm, are called diploblastic animals. Example, phylum Nidaria and phylum Ctenophora. In that ectoderm gives rise to the epidermis, outer layer of the body wall in adult. Endoderm gives rise to the gastrodermis. Endoderm gives rise to gastrodermis. So that we need to check it out. Outer epidermis and inner gastrodermis. This is gastrovascular cavity or gut cavity, we can say. Okay, in between them, we are going to say that mesoglia is present. That gel-like mesoglia is present. Sometimes you may find cells also there, but they are either derived from the ectoderm or endoderm. But generally, there is a non-cellular layer. If present, either they may derive from ectoderm or endoderm. So that's a diploblastic organization. Next. Okay, so we need to identify this in the diagram given here also. That means you see here, this is the ectoderm portion and that inner is the endoderm portion. In between the red color, whatever shown, that is mesoglia, very famous, we are regular in examination. Okay, so that if you see there, the same red color in triploblastic organisms here, that you'll find cells. That yellow, with yellow I am closing here. When I open, you see. You see the yellow portion is open. That means there you will find cells. You can see that a layer called mesoderm. A third germ layer is formed, mesoderm. Now we will try to compare with the both diagrams here. That here the red color, whatever present, that is not with cells. Even present, we say they are from ectoderm or endoderm, they get formed. But as new special layer we are observing here, that inner layer, what the middle layer, what do you say that as mesoderm. We can say that as mesoderm. Let's talk about the triploblastic organization. So before we talk about the triploblastic organization, we'll make it formality to go there. The triploblastica, that address I am giving from the beginning, kingdom Animalia classified into two subkingdoms. Among them, Eumetazoa. Eumetazoa classified into two grades. Among the two grades, grade Bilateria or Triploblastica with what the number of germ layers? Three germ layers. Hence, we called it as Triploblastica. The symmetry is bilateral symmetry and type of organization there. Organ system grade of organization. So, examples. Examples when it comes from phylum Platyhelminthes to Chordata. The total, whatever comes here after these, that this series, total series, phylum Platyhelminthes to Chordata, all are having what type of the number of germ layers in them are going to be that three germ layers, triploblastic condition. We can see that is said to be triploblastic condition. Okay, so now let's go to the text. We'll find and check there. Then triploblastic organization. The animals in which the developing embryo has a third germinal layer, that mesoderm, in between the ectoderm and endoderm are called as triploblastic animals. Example, flatworms to chordate. What is flatworm? Platyhelminthes. Phylum Platyhelminthes. To chordata that I show now. The most triploblastic animals have an organ system level of organization that I shown to you. Tissues are organized into organs which form excretory, nervous, digestive, reproductive, circulatory, and other systems. And triploblastic animals are usually bilaterally symmetrical. That recall the taxonomic grade Bilateria is also called triploblastica. We just recalled it now. So that's it about the triploblastic organization. So that the diploblastic and triploblastic based on the germ layers, we classify it in the form of diploblastic and triploblastic. Okay. Then